Thank you everyone for joining us again. At this time, I'd like to turn things over to our technical chairs for this final session, to Doctors Krista Wigington and John Yin. Thanks so much.
All right, thank you, Emmanuel. We have a great set of talks this afternoon. I want to remind everybody to post questions that they have for the speakers that we will get a chance to answer during the Q&A at the end of the 3 talks. Post those questions that you have in the chat, and those will be archived, and then we'll pick from them in the section following. So I'm going to start by introducing our first speaker. 1 second here. There we go. Professor Denise Kirschner from my own institute, the University of Michigan. Dr. Kirschner received her PhD in dynamical systems from Tulane University, and she conducted her postdoctoral research at Vanderbilt University with joint appointments in mathematics and infectious diseases. She's been a professor at the University of Michigan for over 20 years in the Department of Microbiology and Immunology, where she's now a full professor. Her research focuses on applying mathematical and computational techniques to study questions related to host-pathogen interactions. And a main focus of her research is on persistent infections with pathogens that have evolved strategies to evade or circumvent the host immune system. So with that, I'm going to pass it over to Denise. Thank you so much for joining us today.
Krista, for that introduction, and I'm ashamed to say that Krista and I are at the same campus, but we haven't met, which is just terrible. But we'll rectify that post-COVID and have coffee at Angelo's at some point. So thank you to John and Krista for the invitation today. And I'm going to just wanted to get the pointer going and share my screen. All right, so what I want to talk to you about today is what we consider a systems biology approach to understanding infection interventions. And I'm going to use tuberculosis as my example, but certainly things that we're talking about can be applied to many diseases. And so I want to just start with acknowledgements. I have a large group of fabulous postdocs and graduate students, as well as past members. I know Dr. Pianar was on yesterday, and even some of the work I'll mention today she did, as well as collaborators at many universities in the country and funding from the NIH and Bill and Melinda Gates Foundation. So I want to start by kind of defining what is a systems biology approach. And we sort of think of it as multiple modalities to address a single question. So the way that works is, it will not let me advance. I lost my mouse. What is going on? There we go. So for example, we use monkey models, we use human data, we use in silico math and statistics and computation, and we use microbiological and immunological approaches to understand the intersection of all of these to address specific problems related to disease treatment. And so one of our goals really is to build a virtual human so that we can develop personalized medicine. And we do that over examining multiple scales from molecular to the whole host scale, which I'll talk briefly about today, as well as looking at multiple applications like vaccines, drugs, host-directed therapies, biomarker discovery, as well as something we call virtual clinical trials. So one thing I want to point out today that is maybe a little bit different from the way we've been thinking about things the past 2 days is that there are a contrast between emergent and reemergent diseases. So a lot of the diseases that have been mentioned the past couple of days have been emergent, and those are things like SARS and AIDS, and those are viral-driven pathogens, and they are sort of newly afflicting humans. I'm very interested in reemerging diseases that have been around for a long time, caused lots of morbidity and mortality in the world, and is defined as their incidence is either rising or steady, and their geographical range could be expanding. Talking about things like cholera and tuberculosis, which are bacterial-driven diseases, and malaria, for example, which is a parasitic-driven disease. And so very different pathogens from what we've been looking at, but still as important to think about in terms of prevent. So I'm going to focus on TB today as the example that we apply to look at these kinds of questions. So Before COVID, TB is the #1 cause of death in the world due to infectious disease. 1.5 to 2 million people die a year of TB, and 3 million people die. 3 people die every minute. So during my talk, we'll have 60 people die of TB, this is a huge problem because we don't understand the trajectories, which are either you clear infection, somehow you're exposed and you don't get it. Primary disease, where you really are infectious, and if not treated, you'll die. And latent TB, where you're able to control disease. And you're not infectious. However, over time, there's a risk of reactivation. And that number of reactivation percent can be increased because of things like immunosuppression and drugs or aging. This is a real problem. And so I'm showing you the bacterium and the formation of these structures, which I'll talk about in a minute. So basically, you breathe in the Mycobacteria tuberculosis, and local antigen presenting cells and phagocytic cells take up the bacteria, and they grow very happily inside. But dendritic cells will soon scoot to the closest lung draining lymph node, where it will elicit an important adaptive immune response, where these T cells are then primed and recirculate back into the lung, leading to the formation of things known as granulomas. And you can see here that not only are these T cells able to activate macrophages, which can kill their intracellular bacterial load, which it was not able to originally, but you can see the formation of this structure where really it's a stalemate between bacteria and infection, where you've walled it off, you're containing it, but you're not really clearing it. Although many cases the granulomas do sterilize. So it's incredibly important to realize that most infectious diseases, specifically TB, is a highly multiscale disease. I've talked to you about the granuloma scale, but clearly events at the cellular and molecular scale are involved. Major organs, I showed you the lymph nodes and cells traveling through blood. Of course, there's the whole host, and then what's happening at the population scale. I'm just going to mention briefly, we have an animal model. We have a colony of 300 monkeys supported by the Bill and Melinda Gates Foundation that has a beautiful 50-50 split between active and latent TB that we use to parameterize and validate and calibrate our models. So as I mentioned, we think of TB as multiscale in both space and time. We do something called a mesoscale model, where we start in the middle and then we build out from there. So we put a lot of energy in building a model at the cellular scale that reads out at the tissue scale. And then we consider the involvement at higher scales as well as lower scales. And of course, this all fits inside the framework of the whole host. So just briefly, what we've done here is we've built an agent-based model. Actually, we've built models of all kinds, and we've written papers comparing the different approaches, but we're really comfortable using this agent-based model that has stochastic and discrete elements to it, because it really allows us to track mechanistic interactions between the cells and the bacteria and the molecules like cytokines that are operating all within the lung environment. So if you think of this as 2 millimeter by 2 millimeter lung tissue, you have portals from blood. The lungs are highly vascularized, where cells can enter as well as molecules. And then cells undergo a number of rules and interactions that are all governed by known biology. And so what happens here at the cellular scale, we see the readout of a granuloma. We do not tell it to draw a granuloma. A granuloma is a natural manifestation of this agent-based model. It's called emergent behavior. And so here I'm showing you immunohistochemistry slice of a granuloma from a non-human primate that's been stained to look at different cell types. And here's our manifestation of a computer-generated granuloma. Now, what you can see is that of course, we match, and it's not really necessarily important that we can recapitulate that, except for the fact that they really can only get these once they necropsy the animal, where we can actually track the evolution of these granulomas over time. And so this structure, which has been a veritable black box for experimentalists, now we can watch it evolve over time. We can manipulate it through sorts of knockouts and overexpression studies that are done virtually, sort of predict what's going on. And in particular, we can study things like drug treatment. I'm just showing you this to indicate that while there's a lot of non-human primate data, it's only given to us sparsely. And so, of course, the modeling can predict a much wider and longer range of outcomes that are still consistent with the data that's available. So I want to talk to you about treatment for TB. You know, it's a very complex disease. TB is different from all other bacteria in that most bacteria double 20 minutes, like E. coli. Mycobacterium tuberculosis doubles every 24 to 48 hours. So this slow growth is really its unique virulence factor, if you will. And so it takes about 6 to 9 months with multiple antibiotics to treat TB. Now, most people don't even finish their 10-day course of antibiotics. So you can imagine the compliance issues getting someone, particularly in developing countries where maybe access is limited, taking antibiotics, 4 of them for 6 to 9 months, particularly knowing that there are some side effects. Treatment is long, there's poor adherence, there's drug resistance that develops, particularly in light of non-compliance. These granulomas I showed you are heterogeneous. We'll talk about that in a minute. And of course, patients are heterogeneous. Some people are fast metabolizers of drugs. Some people are slower metabolizers. All these things come together when you want to develop drugs. And right now, there are many drugs in drug trials currently. So if you think about trying to predict what the best antibiotic regimen could be, you have what we define as the regimen design space. So you have these treatment segments. Let's say you can treat over these 6 months in 2-3 month gaps. You have 10 drugs, although there's even more than 10. You can do, numbers of drugs per the segment that you decided earlier. You can vary dose in mgs per gig, and you can also vary frequency. Should it be once a week, every day, every 3 days? If you just look at the possible combinations of this, you get something like 10 to the 17th options for antibiotic regimens. Even with models, that's too complex. So we even have to come up with ways to optimize before we can even begin to solve what the optimization problem really would be here. So we've done that, and we were able to take GrandSim, our original model, and include a plasma pharmacokinetic and pharmacodynamic model that fits in where the drugs entered through transit compartments, depending on the drug, what we know about the PK of those drugs, into the tissues, and track what happens. The killing happens through something known as emax, and that is the rate at which half of the maximal killing is half max there. In terms of each antibiotic, there's a specific Emax for every antibiotic in the system. There's also drug interactions in our model, so we take into account the interactions between these combination drug cocktails. So for example, this just shows you that we're able to track the numbers of doses in a 7-day dose, once a day in a week. You can see how the drug is changing over time and the different bacterial populations depending on where they are sitting in the granuloma. And you can see that there are C50s here, depending on which bacterial class you are. And the drug is spending most of its time below those C50s. So no wonder it takes 6 to 9 months to treat. And more importantly, what we're able to show, and here's a granuloma that we're going to treat with 2 different drugs. One is called rifampin and the other is Isoniazid. And you can see even after a single dose, the RIF is staying around actually until the next dose, but the INH half-life is so quickly it's gone. Notice also that most of the drug in this granuloma is on the outside of the granuloma and not within it. And that's because we talked about the heterogeneity. So you'll see the second dose come on here in a minute. The heterogeneity of this spatial environment, similar to what a tumor would be. This heterogeneity really impairs the drugs getting in, and certainly in this case, getting to these bacteria that could be even trapped in the center in these hypoxic areas. The other thing that we can do is we can apply sensitivity and uncertainty analysis, where we can actually vary the properties of the drugs and actually see, can we do even better with the drugs that we have? So if we take an existing drug like Isoniazid and we vary the metabolism rate or the plasma clearance rate or the cellular uptake just by a little bit, 20%, 50%, or even 90%, can we improve the sterilization, time to sterilization, and the treatment failures? And so we're able to do that. And this allows us to be able to use modeling to really figure out ways to just slightly improve over what we already have. So we've got an optimization pipeline that we've created where we have our granuloma. We look at the PK/PD of whatever antibiotics that we're looking at, and then we have an objective function made up of potentially time to sterilization and dosage. You always want to minimize dosage and minimize time, but you can certainly fit other things into our into our formula here, our objective function, we use something called surrogate assisted optimization to optimize this, which kind of helps you find the maximal on a topo map, if you will. And then when we make a prediction, we test it virtually in virtual clinical trials, which I'll talk about in a minute. And then our best outcome we test in animal models. So I want to tell you a little story. HRZE is the standard for regimen treatment that's used for TB. And recently there was a lot of studies that looked at swapping out the H with moxifloxacin, which is a fluoroquinolone. And so we did this study using these drugs and including moxie. And what we found out was that, yes, definitely when you swap out, you get improvement over the sterilization time from just the standard treatment. But if you swap in it into a different position, look at we can actually potentially get much better sterilization times. So we have a non-human primate study now that's got a series of monkeys that are testing and comparing all three regimens to see if our prediction is borne out. And this would be the perfect thing to do before a clinical trial, to do it in the computation, test it in an animal model, and then move to the human setting, clinical phase 1. So that brings me to virtual clinical trials. There's lots of different ways to do virtual clinical trials. Our goal here is to sort of take things from a granuloma scale, where we have multiple granulomas in a lung, and move to a population scale to really test our predictions. So to do that, we actually need to have more of a whole host model. So that also allows me to talk about vaccines, which requires a more whole host approach. So the vaccines for TB are attenuated live vaccine with something called BCG. And it is given in most of the world, but not in the US or the UK. It has very low efficacy and it's waning protection over 5. So over time, it might be good between 0 and 5 years old, but over that time it tends to wane, which is why there's such a variation here. However, it invalidates the PPD skin test, which we use in these countries. And so we'd rather have a skin test, which works really well, versus a vaccine that's not so efficacious. There are also new tests for TB. The IGRA test tests an immune response, and the BCG can also give mixed results with that.
We have about 5 minutes left, Dr. Kirschner.
And so, so human studies go into phase 1 after mouse studies. Non-human primates are also being used. So the question is, can modeling help decipher differences in vaccine responses and predict better dosing? So a few years ago, Sophie Rhodes and a group of us wrote a paper on immunostimulation and immunodynamic modeling. So let me just review this. The idea is what we do is we figure out the best range in a dose with vaccines, what's the most immunogenic, and then we pass that to humans by scaling up allometrically. So there's a lot of problems with that idea, namely the immune response between the animals and humans are different. The scaling may not be the only thing. It could be nonlinear scaling. And so that may not be the best approach. Of course, we know I've been talking to you about the site of infection, but there's lymph nodes and blood involved. And so we've built a model now called HostSim, where not only do we have lungs that have been, these lungs were taken from CT scans and XYZ coordinates translated. We have our granulomas forming there along with blood vessels, as well as multiple lung draining lymph nodes. And we now have that model up and running to test these things. So for example, we can infect the lungs, and you'll see through the blood the different cell types and how that ends up. And you can see over time the granuloma is beginning to form. So we're in a position now, we call this our whole host model, because in TB with pulmonary disease, most of the story lives in these physiological compartments. Something important that we see is something called recall response. So therefore, after time, if you introduce infection again, you get an immediate and higher recall than you did originally. So what we're proposing is that not only after you get information from the mice, should you do some dose prediction relationship in the mouse, but then you should actually allometric scale that to humans, study it again with models, and then go into the humans for phase 1. And this should cycle through this process here, rather than just having the line go from 1 to 4. The other thing that's been really useful is we've been using our models to predict differences between what we see in human results and, for example, in this case, non-human primate. So there were some beautiful vaccine studies done on a new H56 vaccine for TB that was done in non-human primates and humans. And so we used our model to predict what things made sense to translate between the two and what things didn't. We believe that helps inform clinicians and experimentalists on what's the best targets to move forward in the setting of the vaccine. And finally, we've developed a number of tools to allow us to do these translations: uncertainty and sensitivity analysis, model calibration tools. We do something called tunable resolution, which allows us to coarse and fine grain our models at will, depending on which compartment we're focusing on. That helps with computation. We have parameter identifiability tools, optimization tools, and of course we use machine learning. And what we've done there is really exciting. We'll take limited data sets from animal studies and humans, and we will add synthetic data sets together, and then we will do machine learning on both. So all those papers are available on our website, and I'll just end with a summary of what I think are strengths and barriers of the current approaches. I believe mechanistic models are key to identifying processes driving outcomes. Prevention efforts for diseases are important for those that are endemic as well as those that are emergent. Within host modeling can aid significantly in identifying vaccine targets, drug regimen optimizations, drug development, and drug targets. Virtual clinical trials can speed time and save costs. Right now, there are 17 clinical trials for TB vaccines. Each one cost $1 billion. We've examined them, and we know that at least 5 of them will not work. Modeling can link with host infection dynamics to blood readouts. So people do biomarkers from blood. Modeling can help figure out what's going on inside to that compartment and help with biomarker discovery. Modeling can distinguish between animal and human studies, as I mentioned. But these 3 are really important goals that also can serve as barriers if they're not considered. First, there's got to be collaborations between experimentalists, clinicians, and computational modelers, and grant support to foster that. Considering comorbidities, most of the places where these diseases are endemic, comorbidities are extreme. Even if you think about COVID-19, heart disease in the US was a major barrier in people that were getting disease here, and HIV and TB in Africa were major barriers. And then the other thing is, as you build these more and more complex models, we do need access to high throughput computing options, because we all can't have supercomputers in our universities or in our labs. Yeah, so that's all I have to say. Thank you so much.
Thank you very much. That was really interesting. I'm going to pass it off to John, who's going to introduce our next speaker, and then we'll have questions after our third speaker.
Yes, I have the pleasure of introducing Vipin Kumar, who is a Regents Professor and holds the William Norris Chair in the Department of Computer Science and Engineering at the University of Minnesota. His current research focuses on bringing the power of big data and machine learning to understand the impact of human-induced changes on the Earth and its environment. Professor Kumar's work has been honored by the ACM SIG-KDD Innovation Award, which is the highest award for technical excellence in the field of knowledge, discovery, and data mining. That's KDD. And he's also recognized by the 2016 IEEE Computer Science Sidney Fehrenbach Award, one of the highest awards in high performance computing. Dr. Kumar, we look forward to your talk.
All right. Thank you for the introduction and for the invitation. As you know, my background is in computer science, and my interest is in basically bringing the power of machine learning and big data technologies to environmental problems. And some of the problems that I'm interested in is trying to sort of see if we can understand how the climate, especially the extreme weather, may be changing in response to human actions or natural variations, and more generally how ecosystems like our forests, our lakes and river bodies and the other environmental systems that we depend upon are again changing in response to climate change and again human actions. So we are also trying to make predictions about the future, and I guess that's the commonality. And I was asked to sort of share my experience in bringing the machine learning and scientific modeling together in these contexts, and sort of what lessons could be learned from experience in these domains to the pandemic planning. And I've been listening to the talks since yesterday, and this morning, and I can sort of say that there are a huge number of commonalities, and many issues sort of come up in various talks, including the one just prior to my talk here. It's clear that in pandemic modeling, you're looking at phenomena that's happening at multiple scales, all the way from molecular scale to daily and weekly and seasonal, and of course, population scales. There are multiple disciplines involved. So you obviously require team science. There is yesterday and in the opening talk, it was mentioned that you need to sort of be able to create data from small amount of data. This is again, you're treating data, and going weather forecasting, of course, you see that data being created. There's a need to adapt to data that's becoming available, like data assimilation sort of as an example. And above all, you need to handle the heterogeneity, which is sort of came up in a question earlier today in terms of a concept drift. So all of these are issues actually that sort of come up pretty much in any scientific discipline, and many of these challenges have to be handled. And we are sort of trying to see whether bringing machine learning and scientific modeling together can help us address some of these challenges better than using any one of the technologies by themselves. So with that, I'm just going to it will be a very sort of a brief high-level overview, and I'm here, of course, to learn as much as to share what we sort of know here. So in the sense, if you're trying to model an ecological system, an environmental system, or a weather, or a climate, you're looking at basically some sort of a physics-based model or biophysical model of a dynamical system, which can be represented as a system that has some input, which generally varies in space and time, creates an output, but the system has some hidden states and parameters. And there are some physics-based equations that sort of would at any given time, given the inputs and the state, will create a new output. And also some of those equations would also modify the hidden states. And of course, these may be physics-based equations, or they could be mechanistic models, or some combination of these. And you can sort of see this paradigm unfolding in many, many domains. I'm giving you some examples here from hydrology, aquatic sciences, and fluid dynamics. But if you look at the pandemic planning, you will sort of see something very similar. You're trying to, in the pandemic planning, the output, for example, might be the outbreak, the prevalence of infections and such. But the input would be, again, the parameters that are input would be sort of the situations that are sort of unfolding in the society. Parameter perhaps could be the policy-based solutions. So whenever you have models like these, you're trying to make predictions, and you sort of run into problem in the sense that the models, all models are wrong, some are useful. So basically, the models are never perfect. They always have incomplete representation of phenomena, or certain aspects that are completely missing. And again, these are because of the scale, speed, accuracy trade-off, because you cannot model everything. Even if you wanted to model everything, and even if computation was not an issue, you won't be able to find the data to drive it. So that means you're always making some approximations, for better or worse. And as a result, you have inherent biases that can result in your predictions. And because you have phenomena that you're trying to model at multiple scales, and you can only model it at certain scale or limited set of scales, you model the other aspects using some parameters. And again, you can sort of look at any pandemic model, and you will see many parameters in that as well. But that happens for any physical model as well. And these unknown parameters have to be calibrated, because you have some limited observation data, and then you use them to figure out what value of these parameters really fit those observations. And of course, this is a caricature, but this caricature applies very widely to many scientific situations, especially environmental situations. So for example, one of the parameters, if you're trying to model the basin of a river for hydrology, for its being able to flood in response to rainfall, for example, some of the parameters might involve knowing what the quality of the soil is at different levels. And of course, a lot of those things are not even visible. So you have to estimate what they are from the data. And whenever you're trying to estimate these parameters, it's very easy to overfit them to the observations. And because the reality is much more diverse, much more heterogeneous. And again, you can see many similar situations in the pandemic planning. So one alternative that people have tried to pursue in response to the limitations of the physics models, and because of the excitement that some of these black box data science models have created, especially in the recent years, is to see whether they could be used as an alternative to modeling these dynamical systems. Because if you have data, you could use them, perhaps if you have plenty of data, you could possibly use them directly to go in from input to the output. And as I've been I've been following at least just some of the literature in pandemic planning, and I have seen there have been sort of papers coming up which are sort of trying to make the projections purely from data without having sort of any underlying theory behind it. So people sort of have been sort of doing that for pandemic planning or at least projection as well. So there's a huge tradition of these data science models dating back to many decades. But most recently, for example, deep learning has become an extremely powerful tool, and which has already found hugely successful in many commercial applications and are increasingly being pursued for application in scientific domain. But in many of them, they have been sort of disappointing results because these state-of-the-art complex deep learning algorithms that are very powerful can be very good if you have lots of data. Without that, they can have disappointing results. They can have sort of very poor results. They can create, in addition to that, they can create physically inconsistent results. They can fail to generalize to unseen scenarios. They can, they're unable to provide valuable insights. So in response to this, community, the scientific community, and the people, especially those who are interested in bringing machine learning and scientific community together, have been pursuing this dichotomy between scientific theory-based models and the data science-based model. And if you look at each one of them, theory-based models are limited by our current scientific understanding or our ability to model the phenomena at the right level of details. But the data science models can do really well if you have lots of data, but if the data is underrepresented, which happens the case many times, then they're sort of limited in those situations. So the new paradigm that people are sort of trying to pursue is, can we bring a hybrid? Can we build a hybrid of these two models? Can we sort of bring a data science model which is guided by scientific theory? And this direction, of course, has been pursued by many people. We have been talking about it as far back as 2014. But now, in more recent times, this has become very popular, and huge number of sort of people in different disciplines are following it. Every discipline, you can sort of find some examples of people pursuing it. More recently, just a few months ago, we wrote a survey paper which had about 300 papers on this topic in just last few years. There are research programs and conferences and workshops devoted exactly to this topic. So this is a very vibrant topic. So
just about a minute or so left.
This will be my last slide, and I know I should be short of time here. So the scientific community which is sort of pursuing this ecological forecasting, environmental forecasting, is sort of trying to figure out how you bring the physics or the scientific theory and machine learning together. And they're trying to pursue some of these questions, which I sort of listed them here. And as I listened to the talks over the last 2 days, I can see each of these questions sort of being relevant for the discussion we have seen in the pandemic planning. And there's a lot to learn from what we have done in the environmental sciences and what perhaps could be done in the pandemic planning. Sort of, there's a lot of two-way collaboration that's definitely possible. And just to take a minute to sort of go through these questions, some of the biggest questions that we are trying to pursue here in this quote unquote theory guided or physics guided machine learning modeling is first question is, can we outperform pure physics models or pure mechanistic models or pure machine learning models? That means can we provide better accuracy with less observation data? Can we produce results that are physically consistent? Can we generalize to novel testing scenarios to sort of handle the concept drifts and things like that? Can you handle a complex system that is a collection of processes where the processes are unfolding at different scales? And that happens a lot of times. If you're looking at a hydrological system, the rainfall happens on a daily scale. The soil moisture changes much more slowly. The snowpacks, again, take weeks and months to form and to go away. we had, on the entire country sort of went through this cold snap, and which was apparently caused by the North Pole losing a lot of ice. So these are all processes that are unfolding at with different scales. Can you assimilate new information and data? Because any system has multiple parameters, not just the one that you're trying to predict. And some of those values would become available to you from time to time. And being able to assimilate that information is extremely critical, for example, to produce good weather forecasts. Same thing is, could you assimilate new information as it becomes available, partial information that becomes available in this physics-guided machine learning model? Yesterday, Madhav Marathi talked about being able to create data that for public consumption, which is necessarily at a much higher resolution than what's available from these models. In the machine learning community, people talk about super resolution, going from coarse data to much finer data. Can we make those super resolution methods physics guided so they can be able to handle some of these complex environmental applications? So there's all of these questions that are being pursued in our group, but also in many other groups as well. And I see huge relevance of what's going on here versus the pandemic planning. And I'm looking forward to the discussion in the breakout sessions and of course in the question and answer session. So I'm going to stop here and then again, take any questions later on. Thank you.
Great, thanks, Dr. Kumar, for a very stimulating talk. I'll turn the mic over to Krista to introduce our final speaker.
All right, I'm excited to announce our final speaker. Dr. Rita Colwell is a distinguished university professor at the University of Maryland at College Park and at Johns Hopkins University, Bloomberg School of Public Health. Let's see, her interests are focused on global infectious diseases, water and health. Dr. Colwell developed an international network to address emerging infectious diseases and water issues, including safe drinking water for both the developed and developing world in collaboration with the Safe Water Network headquartered in New York City. And she also served, as many of you know, as the 11th director of the National Science Foundation from 1998 to 2004. So it's an honor to introduce you. I'll pass it off to you, Dr. Colwell.
Sorry about the cover page. Let me start. The previous talks were superb. Denise from the molecular expanding to the most recent talk on the model systems. What I'd like to do, because I think this all fits together extremely well, is to show how we're taking a disease, cholera, and this is a global disease, a water-related disease. We are in the 7th pandemic. In fact, we were in 2 pandemics. We have been in the 7th pandemic of cholera since the 1960s, and then overlaid with COVID. But I'm going to show you, I hope that you will be able to see that what we've learned from cholera, we're able to apply to prediction of COVID. Cholera occurs in about 50 countries, it affects millions of people. We've unfairly labeled the Bengal Delta as the homeland, but 100 years ago, cholera was in the US and Europe, etc. The bacteria, we made the discovery some years ago that the bacteria are naturally occurring in the environment, and they carry out carbon and nitrogen cycling, so they really can't be eradicated. We started our work very early on in the Chesapeake Bay and made a series of discoveries, one of which was quite critical, and that is that the bacterium is associated with a host, which is the copepod plankton in the world oceans, and is distributed throughout the world. In fact, we have isolated Vibrio cholerae from Iceland, where there has never been cholera. So the bacterium exists in the aquatic system. The copepod is its host, and I would prefer to call it a vector. It's a vector that doesn't fly, but it swims. Nevertheless, the bacteria are associated in the gut, on the gill, and certainly on the egg case, which you can see in this gravid female, playing a symbiotic role, probably symbiotic, commensal for sure, in producing a very powerful enzyme that causes the egg sac to break and release the eggs into the water column. We developed very early on, about 25 years or 30 years ago, a crude model of the transmission of Vibrio cholerae from the environment to humans, mainly just unsafe drinking water. the criticism was that we were doing all this work in the Chesapeake Bay and it had no relevance for Bangladesh, which is where cholera is endemic. So in our studies, we started 25 years ago and showed quite clearly that when you take water, as the villagers in remote areas of Bangladesh do, you take the water that's contaminated and direct transmission. And indeed, the model that we enhanced by the work in Bangladesh clearly showed that, yes, there's person-to-person transmission and it's highly dominant because of the very poor sanitation, but the environment is really the source of the bacteria. Now, it occurred to us that at that time we were doing this work very early on. Landsat had been launched, and in the 80s and 90s, it was being used to measure various parameters of the environment. Chlorophyll, sea surface temperature, and sea surface height were three parameters measured by the sensors on the Landsat satellite. And we made this discovery and published this 21 years ago, showing that there's quite a strong linkage between cholera cases in the Bay of Bengal and the sea surface temperature. So this is understanding that first you have this chlorophyll measurement, and then you have 6 to 8 weeks when from the phytoplankton bloom, the zooplankton feed on the phytoplankton. So with this correction, you get this very, very interesting fit. There are a lot of other parameters that have been environmental parameters associated with the outbreaks of cholera: flooding, rainfall, heavy rains, sea surface temperature, as I've just mentioned. Salinity is a very strong factor. And interestingly, the charge was that fecal contamination was directly related, but we found in our work that it's really an inverse relationship, that cholera bacteria are present naturally in the environment, enhanced, of course, by the increased nutrient, so that the numbers become much greater, hence larger numbers of cases. We've improved with improved satellite sensing and with the data we've been gathering, we've improved the model significantly. And so with some of the more sophisticated satellites, we have moved from 25 years ago using Landsat to the present time where we use a variety of satellite data gathered and improved the model that we've been able to develop. Shown here is an analysis that we did of Hurricane Matthew, which traversed Haiti and caused another massive recrudescence of cholera from the 2010 introduction. Well, I'm not so sure it was an introduction, but I'll talk about that in a minute. In any case, what we did was retrospectively for that 2015 cholera resurgence in Haiti, we did a calculation, and on the left is from our more sophisticated model, the prediction of the risk of cholera. And then you see on the right, the actual cases that occurred in 2015. We did an analysis in Yemen. The outbreak in 2017 was the worst cholera epidemic in history, really. Millions died. We again did the analysis retrospectively. The risk map is on the left, blue and red, and then the actual cases on the right. Now, this was picked up by the British Aid Agency in January of 2018. In fact, it was by a telephone call where we were asked by the chap of the British Aid Agency if we would collaborate and we would provide predictions for Yemen, and they would locate physicians, medical supplies, safe water, etc. And so we have done the collaboration. We are able to provide 4 weeks in advance, and this is on a rolling basis, every week or every 2 weeks, depending on whether the risk is high or low. And this allowed us then to provide for Yemen, working with the British Aid Agency, DFID, the British Meteorological Agency, NASA providing funding for our work, and UNICEF. And so this continues to the present. And we provide these risk maps for the British Aid Agency and for UNICEF on a monthly basis. And then we bucket up to every 2 weeks and every week, depending on when the prediction is severe. I'm going to
2 minutes left, Dr. Colwell.
yeah.
Thank you so much.
I'm going to move very quickly to and skip this and go to COVID. With COVID, without going into details, we're all familiar with it. We are able, using a DNA sequencing technique, to identify in sewage samples the COVID SARS-2 virus and its variants. Based on other work, it's been shown that, for example, here, if you read from the top to the bottom, 41 patients, read across, the red line is the throat swab positive, the yellow-orange line shows how long after the virus is released into the stool. And we are currently measuring then using this technique of analyzing sewage. We're able to pick up, as many others are doing right now, this has turned out to be a global phenomenon, but it is an area of predicting health in a community. And furthermore, what we've been able to do is modify the model that we've used for cholera, for COVID, substituting or introducing other parameters such as humidity and dew point temperatures, and using satellite data for movement of populations for tracking. And that has given us the ability then to predict cholera risk. And I'll close here showing that what you have seen in the talks this afternoon is an integration, really, kind of a pathway from the molecular to the space, and how the environmental factors are very tightly integrated with disease, and prediction is clearly possible with the very sophisticated tools now available. Thank you. I'll stop sharing.
Thank you, Dr. Colwell.
Yes.
And, so at this time, we're going to move to our panel discussion. So Dr. Colwell, we'll invite you to stay, and we'll invite all of the prior speakers as well to continue to make themselves available as we address some audience member questions. And so I'll turn it back over to the session chairs for the Q&A.
Great, thank you. Krista, would you like to lead off?
Yes, I will lead, and then maybe we'll take turns. How's that sound, John?
Sure.
All right, so let's see. The first question I have is, I think it's for the first talk for Dr. Kirschner. Thanks for the talk. Could you elaborate a bit on how you merge synthetic and real data for doing ML? Do you treat them as equivalent?
So that's a really good question. And I can point to the paper where we did it, but what we do is try to simulate the exact same study as the non-human primate study, and then generate the same type of data, and then merge it so that it really increases the basically n size of our study, and that gave us the actual statistical significance that we needed to do the machine learning on.
So I can follow up now. This is a question for Dr. Kumar. Do you think TGDS, TGML takes away, and I'll ask you to define those, takes away from the surprise finding side of data science in cases such as pandemic prediction and modeling? Do you think that this TGDS-ML can help, especially since the models could get very complex and heterogeneous with multiple domains involved? And I'll ask you also to help for those who are not in the know what the TGDS, TGML are.
Yes, yeah, I think the TGDS, TGML was sort of a acronym that was sort of being used for the theory-guided data science, just like the machine learning being guided by scientific theory, which are as opposed to building a pure black box machine learning algorithm, because the tradition in machine learning is that you give me the data and I will build the model. language translation, for example, uses very little linguistics theories, and yet you can take your smartphone and point to any language and then anywhere in the world, and it does a pretty decent job of translation. So, so I guess the question is what about theory-guided data science? Is it taking away the surprises? Would you still get surprising results? And I guess this question can be sort of looked at from two different perspectives. And one of them is that if you're constraining your machine learning paradigm by traditional theories, is there a chance that you will not find something surprising because you're always guided by the theories? And my answer to that is that what it helps you find is the missing theory, the missing physics, because you can never have perfect model. You always are making approximations, and if your theory-guided machine learning model is doing better than your mechanistic model, then there are a number of reasons for it. And one of them could be that it's not representing the phenomena at the right level of detail. And of course, that's a hypothetical answer, but a very concrete illustration of this is in the context of work we did with USGS, which is responsible for monitoring all of the water bodies in the US. There are several 100,000 of them. And they try to figure out the behavior of temperature, how the temperature is changing in these lakes in response to climate change and the human actions, because that determines the water quality, the kind of fish that can live in them. And it turned out that the state-of-the-art, the operational model that they were using was much, much worse than this joint machine learning and a machine learning model that did most of the learning from their physics model, and yet then use the data to refine it. And then they could sort of figure out that there were certain aspects of the modeling actually were very weak, and they could potentially improve them. So indeed, yes, you could still get surprises, and you could have these methods produce operational results on a national scale. For example, with USGS, we are sort of talking about replacing their entire set of predictions for all of the water bodies that they sort of do in the entire country using this paradigm.
Oh, great. That's very interesting. Thank you. Krista?
I have a question for Dr. Colwell. You mentioned the sequencing of wastewater for variants. I was wondering if you could, I don't know, share with us any findings from that and how, it compared with clinical data on the variants?
Yes, indeed. I apologize for not having organized the slides. That we had a very serious medical emergency in the family the last couple of days. But the answer to the question is that we've been able to show, particularly working closely with the team in Frederick, Maryland, wonderful team, the public health, as well as the wastewater folks. And over the last several months, we've been able to determine peaks in the numbers of viruses in the wastewater. And it is about 7 to 10 days before you begin to actually see cases appearing in the community. So it's an early warning system for the community to be prepared. In addition, we also now have been storing samples since last June when we started our work, and we are able to identify the variants. So we're now able to detect the presence of variants in the wastewater in these 50 different locations in the state of Maryland. These include not just the waste treatment plants, but dormitories and outlets, the wastewater outlets at assisted living facilities, so that it's possible, as we've been able to show for one of the colleges, that we were able to detect the presence of the virus coming from one dormitory, and then everybody in the dormitory could be tested rather than having to close down the entire campus. So it's become a very useful tool. And my last aspect of it I'd like to emphasize is that I wanted to see this kind of community analysis done for a long time, because I didn't have time to show, but we did a big study with Orange County, California, where they're using wastewater coming in from the sewage plant because of the drought. And we were able to detect in the presence of the incoming wastewater agents like viruses, adenovirus, and cryptosporidium, et cetera. Occasionally they would come up, as a really important early warning system for the community. And I think now that everybody is doing this kind of analysis, that this will become a tool that it should for public health.
That's fabulous. I'll follow up a question for Dr. Kirschner. You mentioned the challenges of allometric scaling in applying mouse or non-human primate data to predict what will go on in humans. Not only, I think this is an issue not only for tuberculosis, but for biomedical science, right? Because a lot of work is done on animal models. And I would wonder what kinds of alternative approaches might you suggest as alternatives to allometric scaling to allow us to use mouse or other data, animal data, to help predict what might happen in humans.
So, and that question goes hand in hand with another one I saw that said, you know, how could the studies I talked about today translate to virus infections? But so TB was the example I used today, but I think in general, you know, the approaches that we did are definitely amenable to any infectious disease. I think we've done similar studies with HIV where we had a blood and a lymph node model. So, you know, similar studies like that could take place for viral infections. So about allometric scaling, the idea is that I think it's a nonlinear phenomenon. So even though, you know, we have little, we have big, and we try to scale it, the idea is that there's this big black box in the middle. And so that's where we believe the modeling can come in. And I think if you did dose response studies in the animal and then fed that to dose response studies in a computer simulation for a primate level model, whether it's human or non-human primate, then I think that you're better suited to predict the dose that's going to work in the human clinical trial. And I think the idea is that I just don't think it's a one step. I think it's a multiple step process. And yeah, so I'll just leave it there.
Great, thank you.
All right, we have another minute and then we'll take a short break. So the last question is for Dr. Kumar. Can fluctuations in animal biodiversity data and their habitat be integrated with climate change data?
Of course, yes. And again, you know, if you have results that you're producing on a global scale, that could sort of be very helpful in many ways. And I'll just give you an example. In some of our own work, we have built a database of about 700,000 water bodies around the world below 50-degree latitude, which is the most complete data set. And its dynamics as to how these water bodies have been changing, growing and shrinking over the last 20 years. And if somebody has a biodiversity data on a global scale, it can be put right next to it to figure out exactly how the changes in these water bodies, you know, if the lakes are shrinking, are animals going away somewhere else to drink water, it could be done. So basically, as some of these methodologies, many of them, you know, motivated by this physics-guided machine learning paradigm, are able to produce global scale results. It creates opportunities for us to merge these data sets with biodiversity things as well. And I'll be happy to talk to you. I'll be happy to talk to the questioner further one-on-one to illustrate this example.
Great, I think we can pass off back to Emmanuel. Thank you very much to our speakers.
Yes, thank you so much. I wish that Zoom had a button for a virtual round of applause, just to properly thank everyone for their contributions. Thank you so much to the speakers for taking the time to be with us and to share from your experience. I know that we've all learned a great deal from this conversation.